Hello everyone and welcome back to Dr. Wasif Academy. In previous lectures, we have officially mastered the world of enzymes from their complex 3D protein structures and the precision of the lock and key model to exactly how temperature and pH can optimize or completely denature them. We have also discussed how various enzymes perform their functions in our body. You now have all the tools and vocabulary you need to ace this topic on your international exams. Today's session is a comprehensive Q&A designed to mirror your final exams. We will be tackling a variety of MCQs, matching columns, and high-level conceptual questions to ensure you're exam ready. Before we start, recall the complete chapter of enzymes to answer the questions and understanding the logical and conceptual explanation. I will show you the question on the screen. Then I shall pause the video, or you can pause the video for a few seconds. Try to solve it by yourself and then hit play to see my explanation and the correct answer. Alright, let's start with our first practice question to test your memory on the basics. Here is the question. What is the chemical nature of all enzymes? Your options are carbohydrates, lipids, proteins, and vitamins. I will give you a few seconds to think about it, or you can pause the video right now to choose your answer. The correct answer is C, that is proteins. Let's do a quick recap of why this is the answer. Remember from the introduction of this chapter, which we established that enzymes are biological catalysts, and nearly all of them are made of complex three-dimensional chains of amino acids. That means their chemical nature is purely protein. But carbohydrates and lipids, no doubt these are vital macromolecules for energy and structure, but they do not form enzymes. And while vitamins are often also helpful as cofactors that assist enzymes, but they are not the enzymes themselves. Now, moving on to question number 2, let's see how well you remember the definitions which we discussed right at the start of this chapter. The question is, which term best describes a substance that speeds up a chemical reaction without being used up? The options are substrate, catalyst, product, and hormone. Now, take a moment to lock in your answer or pause the video if you need an extra second. The correct answer is catalyst. Okay, the correct answer is catalyst. Let's break it down why this is the perfect description. By definition, catalyst is any substance that increases the rate of a chemical reaction while remaining chemically unchanged and entirely reusable at the end. Now, how we can rule out the other options? So you know why they do not fit, because this substrate, this is an input. Okay, this is an input, which is the specific molecule that the enzyme or catalyst actually acts upon. This is the product. Okay, product is, this is the output, that is the new molecule formed at the end of the reaction. And hormone is a chemical messenger used for signaling in the body, not a direct reaction accelerator. So that is the reason the correct answer is catalyst. This is the correct answer, option B. Dear viewers, I am trying my absolute maximum to convey this knowledge with the highest conceptual clarity, and your support really means a lot to me. So if you have not yet subscribed to this channel, then please make sure to hit that subscribe button right now and turn on the bell icon. Join our growing community here at this channel to stay updated with every single lesson, as well as crucial exam tips and tricks that will really help you secure the high marks. Let's move on to question number 3. This one checks your understanding of the different players involved in an enzyme reaction. The question is, in an enzyme catalyzed reaction, what is the specific molecule that the enzyme acts upon called? Now, remember this term, specific molecule. The options are active site, complementary molecule, substrate, and the catalyst. Think carefully about the terminology we used and feel free to pause the video if you want, if you need a moment to answer this question. Here the correct answer is substrate, that is C. Let's quickly review the vocabulary to make sure there is no confusion. The substrate is the actual chemical or a molecule that enters the reaction to be changed. For example, starch is a substrate for the enzyme amylase. Okay, if you remember the lecture 5.2, then here I have explained that how amylase reacts with starch. Okay, the active site, this is not a molecule. Okay, it is the physical pocket on the enzyme where the substrate docks. A complementary molecule. Okay, why this is not the answer? Because this is a descriptive term we use to explain how the shapes match, shapes of active site and shapes of substrate matches. Okay, match. Okay, but it is not the name of the molecule itself. And as we know, the catalyst, this is the enzyme itself, not the molecule being acted upon. So that is the reason the correct answer is substrate. Now let's move on to question number 4. This question focuses on the famous analogy we use to visualize how these molecules interact. The question is, which model is used to explain how an enzyme and substrate fit together? Your choices are the jigsaw model, the fluid mosaic model, the lock and key model, and the synthesis model. This should be quite easy for you to answer because this is chapter of enzymes and we are talking about the model which is used to explain how enzyme and substrate fit together. I hope you have answered this question in the comment box. Okay, the correct answer is lock and key model. Okay, because the synthesis model, this is, think about the physical match between the active site and the substrate and select, you have selected your answer. Okay, the correct answer is lock and key model. As we have detailed in previous lecture, that lecture number 5.1, that this classic biological model perfectly illustrates enzyme specificity, and the enzyme acts as the lock with a uniquely shaped internal active site and only one specific substrate, that is the key, has the exact complementary shape to fit inside the trigger, to trigger the reaction. Now let's quickly see why the other options are incorrect. I am giving you complete explanation to give you complete concept and the clarity that how we can answer these questions, respond to these questions in the final exam. Jigsaw model is not a recognized biological model. Okay, this is not a recognized biological model. The fluid music model, okay, if you remember about cell membrane, okay, then you can easily understand that why this is not the answer. The fluid music model is an entirely different concept which is used to describe the structure of cell membranes. And the synthesis model, okay, synthesis model refers generally to building complex molecules, but it does not describe this specific structural fit. So that is the reason correct answer is lock and key model. Now here is the question number 5. This one tests your understanding of what happens at the end of an enzyme reaction cycle. The question is, what happens to an enzyme after it has catalyzed a reaction and released the products? Your options are, it is broken down by the cell, it becomes part of the product, it remains unchanged and can be reused, and the final option is, it changes shape permanently. Now take your moment to answer this question. The correct answer is, it remains unchanged and can be reused. It should be very easy for you to answer this question. Okay, this is one of the most remarkable features of a biological catalyst. Once the active site converts the substrate and releases the newly formed products, the enzyme does not vanish or break down. Instead, it returns to its exact original shape, completely unaltered. This means it is immediately free to bind to another substrate molecule and start the entire process all over again. Now let's look at why the other options do not hold up. Because it is not broken down immediately. Cells reuse enzyme thousands of times to save energy. Okay, that is the reason it is not broken down immediately by the cell. Okay, and it never becomes part of the product. Okay, because this is not the product. This is products are newly products. New products are formed from this reaction, and this is not the product. And Lastly, it does not change its shape permanently because it is to react with other substrates. Next question is number 6. This question looks closely at the subatomic dance we talked about during the temperature slides, if you remember. The question is, how does increasing temperature up to the optimum, remember this term, up to the optimum, affect enzyme activity? If you will not consider this up to the optimum word, this will be difficult for you to answer correctly. Now take a moment and then answer your question. The correct answer is it increases the frequency of effective collisions, because with the increase in temperature, kinetic energy is increased, and then if the kinetic energy is increased, then the frequency of effective collision also increase, and the result of this effective collisions, result of this effective conclusion collision, sorry, effective collisions is higher enzyme activity. So that is the reason this is the correct answer of this question. Now question number 7. This one tackles the danger zone of temperature and tests your command of precise biological terminology. The question is, why does the rate of reaction drop to zero at very high temperatures? Okay, now remember this thing, drop to zero and very high temperature. Here the examiner is not asking about optimum temperature, he is asking about very high temperature. The options are on your screen. Take a moment and answer this question. The correct answer is C. The enzyme is denatured and the active site shape is lost. Okay. Now let's do a quick post-mortem on why this happens at a molecular level. I have explained you clearly in the lecture 5.2. Here is a quick recap that when temperatures soar past the optimum, the extreme heat causes the atoms within the enzyme to vibrate violently, and this violent shaking breaks the delicate internal hydrogen bonds holding the protein's 3D structure together, and as a result the protein unfolds. It is denatured, and because it unfolds, the active site physically loses its specific shape. Now the substrate can no longer bind because it is no longer complementary, so the lock is broken and the key cannot turn, and the reaction completely stops. Now let's move to the question number 8. This one tests our understanding of pH specificity and how enzymes are uniquely adapted to their shape, to their specific environments within the body. The question is, an enzyme like pepsin works best at pH 2. Okay, pepsin works best at pH 2. What would happen to its activity if moved to pH 7? Now remember, this is, we are talking about pepsin. Okay, we're talking about pepsin, and pepsin works well at pH 2. And what will happen if there is an increase of pH level from 2 to 7? Your options are on your screen. Okay, answer your question, answer this question in the comment box. Here the correct answer is it would denature and activity would stop. The option is C. Okay, let's review the environment where pepsin belongs. As we have discussed in previous lecture 5.2, pepsin is a stomach enzyme specifically optimized to thrive in a highly acidic environment of around pH 2.0, and moving pepsin to a neutral environment like pH 7 is a drastic change. This extreme shift alters the balance of charged hydrogen ions around the protein, disrupting the essential ionic and hydrogen bonds holding its three-dimensional structure together. So that is the reason the protein unfolds, meaning it denatures, and the active site permanently loses its shape. The substrate can no longer bind and the reaction halts. Now the next question is number 9. This question takes a deep look at the molecular chemistry behind denaturation. The question is, Which bonds are primarily broken during the presence of denaturation? Okay, primarily we are asking about the bonds and broken down the process of denaturation. The options are covalent bonds, peptide bonds, hydrogen bonds, or the metallic bonds. Take your moment and answer this question. The correct answer is this one, hydrogen bonds. Okay, let's review the molecular architecture of an enzyme to understand why this is the answer. If you remember, an enzyme's primary structure is a straight chain of amino acids held together by incredibly strong covalent links, which we call peptide bonds. Okay, however, to become a working enzyme, that chain must fold into a precise 3D shape, and that folded geometry is held together by weaker internal hydrogen bonds and ionic bonds. So when an enzyme faces high temperatures or extreme pH, it is these weaker structural hydrogen bonds which vibrate violently and snap. The protein unfolds and the active site is lost. Is the reason? The correct answer is C. Now let's move to the question number 10 and let's tackle it. This question tests your understanding of the exact moment the key enters the lock. The question is, what is formed the moment a substrate binds to an enzyme's active site? The options are a new enzyme, an enzyme substrate complex, a denatured protein, or the final option is a metabolic pathway. Take your moment, answer this question. The correct answer is an enzyme substrate complex, that is option B. Let's review the timeline of an enzyme catalyzed reaction. As we have discussed previously, the process happens in distinct stages. First of all, you have the separate enzyme and substrate. The moment they collide and the substrate docks perfectly into the complementary active site, then they become temporarily linked. This combined structure is called the enzyme substrate complex. It is during this brief moment that the enzyme weakens the substrate's chemical bonds, and then it is converted into the final product. Now the question number 11. This question targets the exact business end of the enzyme molecule. The question is, which part of the enzyme is responsible for its specificity? Okay, which part of the enzyme is responsible for its specificity? The options are protein chain, outer surface, active site, or the amino acid sequence only. So think about which specific region directly interacts with the substrate to determine whether a reaction can actually happen, and then select your answer. The correct answer is active site, C. Let's review why this specific structure is the absolute anchor of enzyme specificity. As we highlighted in our previous lecture, while the entire protein chain and the overall amino acid sequence dictate how the enzyme folds, it is the active site itself, that specialized 3D pocket which processes the unique geometric shape and chemical environment. It is this exact pocket that is perfectly complementary to the substrate. So if a substrate cannot fit into the active site, no catalysis occurs, making this specific region entirely responsible for the enzyme selective nature. Now let's tackle question number 12, which is the last MCQ. This question focuses on a very common exam trap regarding how cold environments affect enzymes. The question is, at low temperatures, enzymes are usually, the choices are denatured, inactive, working at the optimum, or converted into substrates. Now remember, low temperatures. Okay, now think carefully about the distinction uh, between the effects of extreme heat and extreme cold and select your answer. The correct answer is inactive. Okay, the option B. Let's revisit that crucial takeaway tip from earlier of this enzymes lecture. At low temperatures, the enzyme and substrate molecules simply lose their kinetic energy, because with the increase in temperature, kinetic energy is increased, and if the temperature is low, then definitely they will lose their kinetic energy. Then they move very slowly, which means they rarely collide with one another. Because they are not colliding, the chemical reaction stops or slows down to nearly zero. However, the cold does not break any chemical bond, so the enzyme's three-dimensional shape remains perfectly intact. Okay, this is the difference between high temperature effect and low temperature effect. At high temperature effect, this 3D shape is damaged. Okay, it is broken. But at low temperature, there is no change in its 3D shape. It remains perfectly intact. So if you warm the system back up to its optimum temperature, the enzyme will start working perfectly again. Now let's move to the conceptual and logical based questions. First of all, remember this takeaway tip for you that sometimes the examiner asks only to explain, and sometimes they ask explain and describe. They both commands are treated differently. In logic based questions, you will have to pay close attention to the command word, that either this is explain or this is describe. If the question asks you to describe, you simply state what happens. For example, the rate decreases, the rate increases. Okay, this is simply what happens. But if this is asked to explain, you must state why this happens using biological theory. For example, the rate decreases because the active site has changed its shape and is no longer complemented to the substrate. This is the difference between explain and describe command. Most students lose their marks by describing when they should be explaining. Now let's move to our practice session again with a conceptual application question. This is the very classic structured question which you might face on a theory paper. The question is, lizards are ectothermic or cold-blooded. Okay, these are ectothermic or cold-blooded, meaning their body temperature changes with their environment. Now you should explain why a lizard becomes very sluggish and moves slowly on a cold morning using your knowledge of kinetic energy and enzyme substrate complex. Now remember what the examiner is asking you. You should remember these terms: kinetic energy, enzyme, substrate complex. Okay, by using these both terms, you will answer this question. This is a multi-mark question. Okay, I highly recommend pausing this video right here, grabbing a piece of paper and trying to write out a full scientific explanation before you listen to this answer. The answer is, at low temperatures, both enzyme and substrate molecules have low kinetic energy. This means they move very slowly, resulting in a low frequency of successful collisions between the substrate and the enzyme's active site. Consequently, the rate of metabolic reactions like respiration, it slows down, providing less energy for the lizard's muscular activity. That is the reason lizards are inactive in cold morning. Okay. This is a takeaway tip for your exams, that when answering an application question like this one, you will have to look closely at the prompt instruction. The question explicitly commanded you to use the terms kinetic energy and enzyme substrate complexes. Even if your general explanation is excellent, if you fail to mention those two specific terms which I highlighted here, and I have also highlighted here, okay, this one. If you will not highlight these terms, you will automatically lose marks on the mark scheme. And always underline the key terms requested in the question and check them off as you write your answer. Now let's look at our next structured practice question. This one brings together enzyme chemistry and human digestion. The question is, the enzyme pepsin found in the stomach. Okay, asking about pepsin. This is found in the stomach. It has an optimum pH of 2. The enzyme trypsin found in the small intestine. Okay, this one is trypsin. This is found in the small intestine. It has an optimum pH of 8. So if the stomach acid was not neutralized by bile as it entered the small intestine, explain the effect this would have on the digestion of proteins by trypsin. This is also a very classic question, and you can easily respond to this if you remember the effect of pH on enzyme activity. The answer here is, if the acidic stomach contents are not neutralized, the low pH would denature the trypsin enzyme, because the excess hydrogen ions would disrupt the ionic and hydrogen bonds which maintain the enzyme specific 3D shape. This changes the shape of the active site, meaning this is no longer complementary to the protein substrate, and digestion then stops. This is due to the change of the environment. Okay, because the environment of stomach and the environment of small intestine both are different. That is the reason pepsin and trypsin, they both enzymes perform at different pH levels, and if the environment changes, then molecular damage will occur. As a takeaway for your exam, when answering a question like this one, do not just stop at saying trypsin will stop working. Okay, this is not the perfect answer. That only gets you one basic mark. To get full marks on a 3 or 4 mark question, you must give the full molecular pathway, identifying that the pH is too acidic for this specific enzyme and state that it will denature, explaining that the active site changes its shape, and concluding that the substrate can no longer fit. Telling this whole story will give you definitely an A star in this question. Okay. Now let's look at third structured practice question for today. This one addresses a huge misconception that many students trip over in their exams. The question is, a student boils a solution of amylase. Okay, remember, this one is the enzyme amylase, and then cools it back down to its optimum temperature of 37 degrees Celsius. Means first of all it was boiled, and then that is cooled down to 37 degrees Celsius. When starch is added to this cooled amylase, no reaction occurs. Okay, explain why the reaction does not restart even though the temperature is not perfect. This is a very good conceptual check to assess your ability and the understanding of enzymes activity. Okay, you can, if you want to pause this video here for answering this question, you can, but for knowing the explanation of this answer, come back and unpause this video and play it. Let's look at why the reaction remains completely dead. Denaturation caused by boiling is an irreversible change. Okay, once enzyme is denatured, it cannot be reversed. This is irreversible change. The high thermal energy permanently breaks the bonds holding the protein's tertiary structure together. Even when cooled, it is recooled. The amino acid chains cannot refold into the original function active site shape. That is the reason the lock and key fit can never be restored. Okay, no doubt, after boiling the solution, it was again cooled down, but during boiling process, the due to that irreversible change, the bonds are broken. That is the reason after even cooling down the solution, they cannot work perfectly. Now, as a tip for you, examiners like to use the word irreversible on the mark scheme for questions like this one. To secure full marks, you must explicitly state that denaturation is permanent and irreversible. Make sure to emphasize that cooling down only restores the kinetic energy of the molecules, but it cannot restore the shape of the active site. It means there is a change in complementary shapes. Okay, nailing that distinction is exactly how you show the examiner you truly understand the chemistry of proteins. Now let's tackle our final structured application question. This one tests your ability to read an enzyme graph and explain the data using molecular theory. The question is, in an experiment, the concentration of enzyme is kept constant. Kept constant. uh, while the concentration of the substrate is steadily increased. Initially, the rate of reaction increases, but eventually the rate levels off, reaching a plateau. Even though more substrate is being added, suggests a logical reason for this leveling off in terms of active site. Now, first of all, understand what the examiner is asking. He is saying that enzyme is constant. Okay, there is no change in enzyme. Enzyme is constant. the concentration of the substrate is steadily increased. Enzyme is constant, but the concentration of the substrate is changed, that is increased. So what will be the change? This is a classic graph analysis question. Okay, now if you remember the effect of concentration, you can answer the question. This, the logical answer here is the rate levels of, because the enzyme concentration has become the limiting factor here, because enzyme is constant, no doubt the concentration of the substrate is being increased, but enzyme number of enzyme concentration is not increased, that is that is constant. So being constant, enzyme is a limiting factor here. And at the plateau, every available active site is occupied by a substrate molecule. This is called saturation. Even if you add more substrate, there are no free enzymes to process them until a previous reaction finishes and the products are released. So that is the reason here. Okay, enjoying this lecture? Then tell me in the comment box. Okay. Exam tip here: when examiners ask you to explain a plateau on a substrate concentration graph, they are looking for three specific keyword phrases. All active sites are occupied or full, the enzyme is saturated, and this is the limiting factor. Okay, remember these terms. If you use these three phrases, you will easily score maximum marks on any data interpretation question. To finish up our practice session today, we have a master matching challenge, which we have where we have three columns here. Column A list our key biological terms, column B gives their description or function, and column C identifies their specific substrate or location. Your task is to match each term from column A with its correct partner in column B and column C. This is quite a puzzle. So definitely you will have to pause the video right here, take a piece of paper and map out your connections. Let's see if you can get all five perfectly aligned. Now let's solve this master matrix step by step and reveal the correct combinations. First of all is about amylase. Amylase matches with C. Okay, amylase matches with C, that is breaks down starch into molecules and into maltose. Okay, and then it is with starch. If you remember, amylase works with starch. So this is the combination. Okay, amylase with C, and then that is with 2 of column C. Now let's look at protease, that is pepsin. Okay, it matches with, protease matches with A, that is breaks down proteins. Protease, remember with this term, protease is effective with proteins. Okay, and then this is found where? This is for pepsin is found in stomach. As we saw in our pH question, if you remember, pepsin is our stomach specialist that thrives in highly acidic conditions to tear down dietary proteins. Third one is catalase. Okay, catalase matches with the D, means it breaks down hydrogen peroxide into water and oxygen, and then this D, it matches with number 3. Okay, it matches with number 3 because this is found in all living cells, for example liver. Okay, catalase, this is an incredibly fast intracellular enzyme which protects our cells by instantly destroying toxic hydrogen peroxide byproducts. Okay, and the fourth one is lipase. Lipase matches with E because it breaks down lipids into fatty acids and glycerol. Remember, lipase and lipids. Okay, this lip is common as a protein, protease and protein. There are common letters here. Also lipase and lipid, there are common letters, so you can easily identify the lipase is effective to break down lipids into fatty acids and glycerol. Then this E, that matches with number 5. Okay, that matches with number 5 because no, this is not matches with number 5. uh, it matches with number 4, fats and oils. Okay, because lipase is effective for lipids and that matches with lipase. Now let's look at question number 5, which is about active site. Okay, active site that matches with B, because active site is the specific region where our substrate binds, and that this definitely requires complementary 3D shape, because the active site is not an enzyme itself but that crucial 3D pocket that matches perfectly with the substrate like a log and a key. I am very hopeful that you have clearly understood about these enzyme functions and their location, and you have matched these columns. Okay, if you have any question or any query, you can freely ask in the comments section. And there you have it, everyone. We have officially mastered the world of enzymes, from their complex 3D protein structures and the precision of the lock and key model to exactly how temperature and pH can optimize or completely denature them. You now have all the tools. Okay, you can easily answer the question in the exam section. Now here is a quote, motivational quote for you, that just as an enzyme is the spark that turns a slow process into a vital reaction, your curiosity is the catalyst and turns information into wisdom. Do not just sit in the beaker of life, be the change that moves it forward. So my dear students, if this detailed breakdown, this detailed chapter has helped clear up your doubts today and in the previous lectures, please hit that like button and subscribe to this channel, and do not forget to click the notification bell so you never miss a lecture, as we systematically cover the entire biology syllabus. Now at the end, if you have any question, then drop that in the comments below. I will read and reply all of them. Thank you so much for learning with me today and joining this question and answers Q&A session. Keep studying smart, stay curious, and I will see you in our next video lesson. Thank you and goodbye.